Look at this simulated microscopic environment. Thousands of molecules and structures are aggressively colliding in a dense, chaotic fluid space. If a cell actually operated like this disorganized free-for-all, the results would be fatal. Vital hormones would drift aimlessly, and highly acidic digestive enzymes would leak out and dissolve the cell from the inside out. This topological map outlines the cell's solution, the endomembrane system. It is a highly integrated logistics network made of phospholipid bilayers, the exact same flexible material making up the cell's outer boundary. Notice the two highlighted pathways. This system manages two opposing challenges simultaneously. It must route newly manufactured supplies outward while securely capturing and neutralizing inbound hazards. A cell requires a massive inventory of chemical ingredients to survive. But those ingredients are entirely useless without the physical infrastructure to route them and prevent absolute metabolic anarchy. The start of this supply chain sits right outside the nucleus, in a sprawling factory known as the endoplasmic reticulum, or ER. One section is called the rough ER because its surface is studded with ribosomes. These tiny molecular machines read genetic instructions to build proteins, folding them step by step into complex, three-dimensional shapes. Once folded, those nascent proteins are pushed directly into the ER's lumen, the hollow interior space of the membrane network, where they undergo their first round of quality control and processing. Right next door is the smooth ER. It lacks those ribosome workers entirely, meaning it doesn't build proteins at all. Instead, it synthesizes lipids for cell membranes and neutralizes toxic chemicals. Physical form dictates function on the factory floor. The presence or absence of ribosomes strictly determines whether a stretch of membrane builds structural proteins or processes chemical lipids. Watch how the membrane behaves here. A section of the ER bulges outward, safely encapsulating the newly built protein, before snapping off to become a free-floating, sealed bubble known as a transport vesicle. This vesicle acts as a cellular delivery truck. It floats through the fluid cytoplasm until it collides with and fuses into the cis face, the receiving dock, of the Golgi apparatus. Inside this cross-section of the Golgi, the payload moves layer by layer through flattened sacs. At each stage, specialized enzymes modify the protein. Focus on those geometric shapes attaching. Through glycosylation, the Golgi adds sugar molecules, acting as precise molecular barcodes. Without those chemical shipping labels, the cell would dispatch its products blindly into the void, rendering the entire manufacturing effort completely useless. When the fully modified cargo reaches the far side of the Golgi, the transface, it gets packaged into new vesicles. Here, the logistics network splits down two primary paths. The first is the secretory pathway. Outbound vesicles travel to the cell's outer boundary, fuse seamlessly with the plasma membrane, and release their contents into the body to be used elsewhere. The second path handles destruction. Watch this white blood cell wrap its outer membrane entirely around a rod-shaped bacterium, swallowing it whole into an internal pocket called a vacuole. To deal with that trapped pathogen, the Golgi dispatches a specialized vesicle called a lysosome, heavily loaded with digestive enzymes. The lysosome docks with the vacuole and obliterates the invader. Because lysosomes maintain a highly acidic internal pH, those aggressive digestive enzymes only function inside the vesicle. By compartmentalizing the acid, the cell isolates dangerous chemical reactions away from its own fragile machinery. The vesicle proves to be an incredibly versatile tool. The exact same phospholipid structure serves as an outbound delivery truck and a secure containment unit for hazardous waste. Zooming out, this wide view shows the entire endomembrane system in motion. Thousands of particles flow from the ER through the Golgi and out to the boundary. Color overlays reveal the system executing constructive cargo exports while simultaneously managing destructive inward containment, completely without error. The efficiency relies on the membrane material itself. As vesicles pinch off from the ER and fuse with the Golgi, or leave the Golgi and fuse with the cell wall, the phospholipid bilayer is constantly flowing and recycling. Every time you digest a meal, fight off a cold, or flex a muscle, you are relying on this exact microscopic infrastructure, delivering hormones, enzymes, and proteins to precise locations. Having the right chemical ingredients is a prerequisite for life. But survival requires the flawless, continuous logistical execution of routing those resources exactly where they need to go.